I think many of the people here and our guests here today would be shocked to learn, and I'm still shocked every time I hear this statistic, that Alzheimer's is the 6th leading cause of death in the United States. What is it, and why do we have to die from it? Dr. Cummings?
Yeah, so I'll start, and thank you for having me here today. It's really great to be in an organization that has had such tremendous impact. I'm a great supporter of Howard and of ADDF, and I'm meeting Leonard for the first time today, and I see why there's so much enthusiasm. So it's fantastic. Congratulations to the Sandlers on taking a great step for this, so thank you for that. But Alzheimer's disease is an age-related disorder in which there are changes in the brain, accumulations of proteins that lead to the death of nerve cells. And all of our mental life is dependent on those cells. Our memory is the history of who we are, and it's our own biography. So as we begin to lose those cells, we begin to lose the sense of who we are, who our own history is, who our biography is. And that's what Alzheimer's disease represents, is the gradual erasing from the most recent to the most remote of who we have been. And that's the process that we're trying to intervene in, is that death of the nerve cells over time that defines who we are.
So why is it so hard to stop that process? Either one of you. You're friends.
Yeah.
I don't want to get into an ego thing about who gets more attention here today.
We wouldn't be looking at each other this way if the answer were easy. And of course, thousands of scientists are working on this problem, and they are underfunded, and there are too few of them, but there are many people who are working on these issues. It's a tremendously complicated disease. It's embedded in aging, and think about how we're all changing in so many ways with aging. And then you superimpose on that another set of conditions with the accumulation of 1 or 2 or 3 different kinds of proteins and many ways of injuring the cells. And we don't know which one of those might be the key. And increasingly, and I was so happy to have this discussion with your group recently, Howard, we think that one drug might not be the answer. How likely is it that one drug could intervene in a whole network of processes? I think it's much more likely that we need to give serious thought to what are the optimal synergistic combinations of drugs that would help us intervene in this process. And we haven't taken that step, Paula. So we need to take the next step and acknowledge the complexity of the disease and then try to respond to it.
Well, certainly Randall and Liz and the Lauders and a number of people and today in this room are taking an enormous step to help you find those answers. Yes. Maybe you can describe to us some of the other challenges in this drug discovery pipeline, which of course is what we've explained to you is the focus of ADDF and its work.
Right. Well, just discovering drugs is a very different science than the biology that we usually think of in basic research. So basic research is an attempt to gather knowledge about how a disease happens. And I truly think that today we know as much about how Alzheimer's disease happens as we know about heart disease and cancer. We've had drugs for heart disease since the 1950s that lower cholesterol. We've known about cholesterol as a risk factor. Heart disease remains the leading cause of death in the United States. So I just want to put Alzheimer's disease in a little bit of context. If we're trying to develop antibiotics, basically we take something from the soil, throw it in a test tube with some bacteria. If it kills the bacteria, we're on the road to developing an antibiotic. We got to make sure it's safe. We have to make sure that it gets into the body and this kind of thing. For Alzheimer's disease, we have all the challenges of developing drugs. The chemistry is extraordinarily difficult. It's a completely different science than the biology. We need pharmacologists who can help us to design these drugs in ways that it gets in the body, it gets in the brain, it crosses the blood-brain barrier. And as Jeff said, the design of these chemicals, because that's what drugs are, is very difficult, and especially when we have a disease where we all think that there are multiple ways that the neurons are getting killed. So that's why I think one of the really important strategies, if you think of a funnel, is that we have many different ways to kill brain cells over time as we age and so on. And if we could design drugs that are neuroprotective regardless of the source of the injury, then we might have a way to develop one drug that could always protect the nerve cells from damage regardless of whether it's oxidation or misfolding or low oxygen and so on. And so that's one of the strategies that ADDF is funding, neuroprotection. And we could talk more about that. But I think, generally speaking, it's hard. We've seen a lot of the pharmaceutical companies getting out of Alzheimer's disease. Not only that, they're getting out of psychiatric illness. They're abandoning their programs because, from a commercial point of view, it's so hard. And that's why ADDF and others and foundations like ours are so necessary to keep the ball rolling.
And you've had a great deal of success in doing research on repurposing of drugs. Can you share with us what some of the strides that have been made?
Yeah, sure. I'd love to talk about our particular repurposing program, which is bexarotene. Bexarotene is an example of repurposing, which means taking a drug that was approved for one condition and looking at it to see whether it would work in another condition. And you do that because there are good, sound scientific principles that would make you ask this question.
Isn't it true Viagra was one of those drugs?
The Viagra was one of these drugs.
We had talked about that. I wasn't going to let you get away with not talking about Viagra.
It became one of those very successful drugs.
Sorry, I asked. But it was actually looked at in treatment of heart disease, right?
That's right. It was originally looked at for congestive heart failure. Of course, it failed in that, but the men wouldn't give it back at the end of the trial. So Pfizer, being a good observer and business company, decided, well, maybe there's something to look at more closely. And the interesting thing about Viagra is it's now been approved for yet another repurposed indication, which is pulmonary hypertension. So Viagra is an example of a drug which has had now 2 fully approved lives in different diseases. And that's what we're trying to do. So the trial we're doing at the Cleveland Clinic, for example, is there was an experiment with a cancer drug that had a dramatic effect on the mouse model of Alzheimer's disease. So it actually reduced one of the proteins we're interested in by 75% in 6 days. Well, that's an observation worth following up on. But I could repurpose that because it was already approved for cancer. That means that rather than having to take it all the way through the early stages of the drug testing in normal volunteers and all of that, it's ready to go. And this cancer actually occurred in older people. So it was also ready to go in older people. So we have started the first clinical trial of bexarotene in the country. We're about halfway through that trial at this point. We're using the latest biomarkers. Howard talked about the critical role of the ADDF in developing a new test that measures the protein in the brain, a kind of brain scan. So that's what we do. We take this exact brain scan that ADDF helped develop. We do it at the beginning of the trial. We then give the drug. We look at that scan at the end of the trial to see whether we've had an impact in removing the protein. Until this scan was approved, there was no chance of measuring that kind of precise impact of the drug. So this is a great example, I think, of repurposing a compound for Alzheimer's disease.
And there is a lot to celebrate when it comes to ADDF and the success you've had with your drug research. I want to give you a chance to share some of the highlights of your most recent victories.
Right. Well, we've seen about 3,000 ideas for new drugs in the last 14 years from all over the world. And we've been able to fund about 400 of them. And a number of programs that we've funded have moved from, as Jeff mentioned, the preclinical state where it starts with chemistry and then goes into animals. But a real success is when drugs get into humans and they start being tested in humans because we have to do the testing in humans. And so we've had more recent successes in phase 2. We're also very much using the repurposing strategy. And we're very interested in repurposing drugs for diabetes and hypertension for Alzheimer's disease. As I mentioned this morning, the brain is totally dependent on glucose for energy. So the brain represents 3% of the body weight and uses 25% of the energy. that's necessary. And there is insulin resistance in the brain. So we've looked at a number of ways of trying to use drugs for diabetes for Alzheimer's disease. So we funded a clinical trial at Columbia University, Jose Luxinger, of metformin, which is the most widely used drug for diabetes in the world. It's off patent now, so it's generic. So there's no drug company that's really ever going to test metformin for Alzheimer's disease. And we helped Jose to design the study, and he just reported the results. And it's looking like, by reducing insulin resistance and improving glucose utilization in the brain, that metformin may have an effect. Now, that was a small phase 2A trial. Again, these financial hurdles at every step of the way, the gap financing. Now Jose needs to do a much larger study. Now he needs more than the $1 million that we gave him for his study. He needs $20 million to do a larger study to prove this. And we've helped a biotech company that just reported results at our annual meeting, in our 14th annual meeting, of another drug for diabetes that's still in the preclinical phase. A biotech company that couldn't get any funding for Alzheimer's disease. They were very far along in their diabetes program, and their board wouldn't let them get into diabetes because they didn't have the money, and they wanted them to focus on that. We gave them money for the Alzheimer program. First, they proved that their drug worked in animals, and then we helped them to design a clinical trial, and they just reported again that this drug improved glucose utilization in the brain of people with Alzheimer's disease. I think another thing that we're doing is using drugs for hypertension for Alzheimer's disease, and that's really important because probably of all the diseases, hypertension is the biggest risk factor for dementia, for Alzheimer's disease that we know of right now. And so there are actually drugs, that antihypertensive drugs, that are actually neuroprotective. In other words, they protect brain cells. So we've just funded a clinical trial at the University of Toronto to do that. And we have other drugs that are moving into the pipeline. We started a company out of Stanford University about 10 years ago. Dr. Frank Longo, who's the chairman of neuroscience there, had an idea that he could design neuroprotective small molecules that would mimic the effect of our natural hormones that are large, and they could never be drugs because they could never be taken by mouth or injected, and they'd be much too expensive to even synthesize. And he screened on a computer 2 million compounds. He found 6 that fit into this receptor. He started testing, and he found 1 that worked in animals, and we've just funded him to move that from animals into human beings. So now he's done safety studies of this. And so, you know, in science, things are incremental. We haven't gotten to the cure yet, but those are just a couple of examples of how we move things forward. We're going to be very excited to see Frank's company that he spun out of Stanford, that we helped him spin out, move into the phase 2 and see if the drugs actually work.
So you're really talking about developing drugs on a bunch of different tracks. You're talking about slowing down the progress of the disease, stopping the process altogether, and then finding a drug ultimately that will cure you if you get Alzheimer's?
Yeah, I mean...
I didn't major in science.
Yeah, I mean, I actually think, you know, I think that it may be hard to get a complete cure just because, as Jeff said, there's not one cause. But I'm very optimistic that I think we'll get effective treatments. And the clinical goal really is to use the kind of test that we help to develop to find people who have the disease in their brain before they become symptomatic. Just like we find people that have high cholesterol and start treating them before they have a heart attack. We can, with this new brain scan, identify people that have the disease before they lose their mind. And we'll develop drugs that first treat people that have the disease, and if they're effective, it's likely that we'll then be able to use those for prevention. So the goal is to find people early that have the disease and slow the course of their progression to clinical symptoms. And there was one study that showed if we could just delay the onset of Alzheimer's disease by 5 years, and the average age of onset is 78, so if we could delay it to 83, we would reduce the number of people that suffered dementia from Alzheimer's disease by 50%.
Wow.
And I think that's an achievable goal.
Well, that ought to be something that we should think about as we leave this room this afternoon.
No, it's a great point, Howard, that since it's a late onset disease, if you just delay the onset, you eliminate much of it. But what I was thinking was you were asking about multiple targets, and maybe it's important to get this idea of multiple shots on goal. And that's the philosophy of our program in terms of clinical trials, is why would we be testing many drugs rather than just, you know, the one greatest drug? It's because you don't know which one is the greatest drug. So you test multiple drugs and then eventually you get one to succeed. But our science is still not well enough developed that we can predict which of these compounds is going to succeed, or perhaps which combination of compounds is going to succeed.
I'm going to backtrack a little to the onset of the disease. I think one remarkable advance that has been made is the fact that you can actually diagnose Alzheimer's, which was something you couldn't do, what, just 5 years ago?
5 years ago.
Tell us how that's done. I mean, there are many, many lives in this room impacted by Alzheimer's, and we all want to know how you find out you have it or whether you might get it.
Yes, I'll tell you about the scan, and this was a scan that ADDF played a critical role in developing. But in Alzheimer's disease, you get the plaque, right? This is the amyloid plaque. That's one of the characteristic features. But we could never know who had plaques and who didn't. And we knew that we often made mistakes in diagnosis, that people we would call them Alzheimer's disease, and then at autopsy or later in life, it would turn out they would have something else. So how do we correct the diagnostic error? Well, a scan was developed on the basis of tremendous research that allows us to visualize the scan, visualize the plaque on a scan in the living person. So now if somebody comes in with a dementia and they have plaque in the brain, we know that's Alzheimer's dementia. If somebody comes in with mild impairment, and half of these people will have Alzheimer's disease and half won't, right? So that's a very different life trajectory with and without Alzheimer's disease. How do we sort that out? With this scan. Because even in the mild cases, the scan will already be positive and plaques will already be in the brain if the person is going on and has the trajectory of Alzheimer's disease. Now, the ethical conundrum. Every new discovery raises new questions, and the ethical conundrum is it's also true that people without symptoms have a positive scan for about 10 years or 15 years before they're going to get the disease.
It's amazing.
Now, that raises a whole new set of questions. People don't necessarily want to know because, of course, there's nothing that we can do at this moment. On the other hand, it opens up the capability of doing clinical trials on those people to prevent Alzheimer's disease. And we have 2 prevention trials, Paula, that we're doing at our center right now, where we're taking cognitively normal people and randomizing them to drug or placebo, with the outcome being, can we prevent Alzheimer's disease? How great is that? How can we prevent Alzheimer's disease? That's great, you know, that's such an important question. And we're finally doing clinical trials in which that's the outcome. How fantastic.
Well, how fantastic is it that we get to sit with 2 of the very men on the front lines that are probably going to answer that for us. Just very briefly, because I know people have to run back to the office and home and everything they have to do. Can you leave us with a 30-second thought apiece about some of the things we can do? I know some of you heard this in the earlier discussion, but for those of you that didn't join us at the symposium, besides exercise, we know the brain shrinks if you don't exercise. What else are some of the things that we can be doing proactively at home that might slow down either the progression of the disease or stave it off altogether?
Well, I think the soundbite that I like to give is that it's looking like, and we have a lot of research on this now, that everything that we've been taught to do to prevent heart disease probably prevents Alzheimer's disease. So if you think about all the forms of heart disease, atherosclerosis and so on, hypertension, diabetes, these medical factors, obesity is a risk factor for Alzheimer's disease, not only perhaps because of insulin resistance again, but we know that that big round thing around your waist is not just a load of pounds, but is actually an inflammatory organ. And we know that inflammation has a very negative effect on the brain. Another thing that I'll mention is we've recently funded a study at the University of California, San Francisco, where we specifically wanted to address the issue of surgery. Because even back in 1980, when I was taught first about Alzheimer's disease, we were always taught that a lot of times people go to surgery and they come out of it and suddenly they have Alzheimer's disease or suddenly they're demented. But there was no way to predict who those people might be. So surgery, major surgery, can be a risk factor for developing dementia, but we'd like to identify those people early. So we use the brain scan that ADDF helped to develop to test people before they went into surgery, to see who was at risk for cognitive decline after surgery. And we've found that the people who have a positive scan are the people who will be at risk. So it's going to help people decide whether or not to have surgery or what kind of surgery. I think that's very important. And I'll let Jeff go on to the next.
Well, I think exercise is very important. The other thing that I would identify, Paula, is Mediterranean diet. We know the diet's very important, so that's a high fish, high fruit, high vegetable, high nut vegetable, high nut diet content, delivered in a social way, right? You got to discuss Berlusconi, you know, with every one of these meals. So
La dolce vita.
It's not only the diet, it's about the social interaction and the learning and the exchange that takes place. And I think that emotional engagement and maintaining the intellectual function complemented the diet and the exercise is how we're going to reduce our individual risk for Alzheimer's
Well,
disease.
it has been a privilege to have both of you join us here this afternoon.
Thank you.
Continued good luck.